Good morning, everybody, and welcome to this week's version of Micro Seminar. I'm very happy to introduce today Dr. Céline Lavergne. She is currently a senior researcher at the Centro de Estudios Avanzados and Hub Ambiental UPLA of the University of Playa Ancha, which is in Valparaiso, Chile. Prior to her current position, she did postdoctoral work at the Pontificia Universidad Católica de Valparaíso. She did her PhD, which she received in 2014 at the University of La Rochelle under the supervision of Christine Dupuy and Hélène Agoge. And she did undergraduate work at the University Blaise Pascal. Pardon me for my pronunciation. Today, she's going to be getting into some very interesting topics, including methane cycle in the Southern Hemisphere and the effects of temperature, as well as the global importance of bacteria in the Mercury cycle. So without further ado, I will turn it over to Dr. Lavergne and take it away.
Thank you, Cameron. Okay, so yeah, I will share my screen. I guess you can now see the screen perfectly.
Looks good.
Perfect. Thank you so much, Cameron, for this introduction. So let's talk about some stories from the south, and from the very south, actually from the Patagonia, Cape Horn, and Antarctica. So as Cameron said before, we will talk about methane cycle and also the importance of microorganisms in the Mercury cycle. So let's begin with the methanogenic pathway and communities in the south of Chile. Here in these kind of beautiful ecosystems, like here you have a peatland just around a beautiful lake. So the first work that I will talk about today, it's in the frame of a big project financed in part by the European Union. This project is called Methanogenic Biodiversity and Activity in Arctic and Antarctic and sub-Antarctic ecosystem affected by climate change. So here it's a collaborative work with more than 25 members from 7 countries, but actually I will speak specifically of one part of the work in Patagonia with the special implication of Paulette Aguilar and Léa Cabrol. So actually, the increasing of temperature at the Earth scale can increase also the microbial activity, and it can have an impact on the greenhouse gas emissions. That can also have a direct positive feedback on, or indirect positive feedback on the global warming, and particularly the methane is of a special concern because of its power in the global warming actually. And methane is coming from both anthropic sources and natural sources, and among the natural sources, freshwater ecosystems account for 43%. Freshwater ecosystems are an important source of biogenic methane. Actually, it comes from the degradation of organic matter, and it's the result of different, many different processes, from hydrolysis to methanogenesis. Here it's that like a global view, a simplified view of this reaction. And what was interesting for us is the limiting part, which is the methanogenesis. Methane can be produced from different methanogenic precursors, such as methanol, TMA, hydrogen and carbon dioxide, or acetates. And actually, what we know is that it is reported that in freshwater ecosystem, methanol and TMA is not the main precursor that the methanogens can use. So the most important methanogenic precursor in freshwater ecosystems seems to be hydrogen, CO2, and acetate. So we will focus on that two precursor. And actually what we know is that organic matter quality, temperature, and pH are the most important factor to be considered that can influence the proportion of methanogenic precursor that they can be used, so acetate or H2CO2. Actually, we will work and we will talk about southern ecosystems. Why we are studying South and Patagonia ecosystems? Because Patagonia harbors unique ecosystems that cannot have any latitudinal equivalence on Earth, as you can see here between these two high latitude 40 and 60 south. So the objective of this first work was to evaluate the temperature-induced changes on microbial community structure and on methanogenic pathway in order to understand which pathway can be the dominant in the actual condition and maybe in an increasing temperature scenario. So here I was used to work in Valparaiso in Chile, and the ecosystems are located in the south in Patagonia. We were working with 5 freshwater sediments of 5 freshwater ecosystems. A reservoir here since close to the Punta Arenas city. This is one of the cities most in the south of Chile. We were working also in a lake with a glacial origin. And we also studied some beautiful and shallow lake or peatland lakes in Cape Horn here near Puerto Williams. So there we measure some physicochemical parameters such as the organic matter concentration, nutrient pH, temperature, etc. And what we were interested in was to measure the methane production rates in these freshwater sediments where methane is supposed to be produced. We use microcosm for that, in which we measure the pressure and the methane concentration by GC measurements. We performed 3 different measurements or 3 different experiments. One experiment without any substrate addition. And that experiment, we did it at in situ temperature and in situ more 5 degrees. And then we added a substrate, some specific substrates known to stimulate methanogenesis. So whether acetate on one part and H2CO2 in the other parts, and that we set it up at 5, 10, 15, and 20 degrees. Then, actually, in the in situ samples, and also at the end of the experiment, we studied the microbial diversity through 16S metabar coding that we analyzed using the tool frogs implemented in the Galaxy platform, and then we performed all the multivariate analysis in R. So here is the objective. Let's go first to the results, looking at the effect of temperature on the unamended experiment. Here you have the result from the 2 first freshwater ecosystems near Punta Arenas, and the value of the methane production weight here. And here you have the result from the south, and what we saw is that an increase of temperature by 5 degrees significantly increased by a factor 2 the methane production. Actually, when we incubated the same samples under this under amended condition, adding acetate from 5 to 20 degrees, what we found is that the methane production rates were positively affected by temperature. And if we have a look on the methanogenic diversity, here we were working, looking only at the archaeal sequences. And here you have an example. So for example, here you have the in situ community composition, and here the incubated community composition. And what we saw is that adding acetate selected methanosarcina in the experiment. Methanosarcina is a pretty versatile methanogen, and it can be surprising because methanosides are also able to use that, was not maintain its proportion more or less at the end of the experiment. Hydrogenotrophic production or methane production was also affected, positively affected by temperature. Actually, adding hydrogen and carbon dioxide was less efficient by producing methane. And when we look at the composition, here you have an example of the methanogenic community composition at in the in situ samples and at the end of the experiments. And what we saw is that methanobacterium can be selected and be dominant in that ecosystems or in that microcosms, but after 15 degrees here, for example, in that sample. What we were interesting to understand is the temperature dependence of that of the different measurements that we've done. So we calculated the energy of activation of methanogenesis in the 3 different experiments that we have done. We calculated that energy of activation that represented the temperature dependence in each ecosystem, and then make the average to obtain that value in electron volts. And we compare it with a previous study from Yvon Josh and collaborator. This study was really interesting because they gave the proof that if we are looking at the temperature dependence of the methanogenesis at the population level, or of the methane production rates at the community level, or the methane emission at the ecosystem level, we can obtain a similar value of energy of activation ranging between 0.8 to 1.27 electron volts. So the temperature dependence at each level was, it can be resumed in a universal energy of activation. Well, actually, what we found is that without any amendments, we are measuring actually energy of activation within the published range. And so do I, and this is the same with a stimulation of methanogenesis by acetate, but we have to nuance these results when we stimulated the methanogenesis with hydrogen and CO2. If we are looking at the whole microbial community now and representing the results on a non-multidimensional scaling here on the left, what we saw first is that the most important was the origin of the ecosystems. So we have data from three to five different ecosystems here, and actually, well, the temperature did not have any significant effect on the whole microbial community structure. And the most important was the origin, the ecosystem where they come from. And then in a second importance, the overdriving factor was the substrate or the methanogenic precursor that we added to the experiments. Actually, the most important taxa that we can find and that were important on building this ordination were all not strictly, but all facultative or obligate anaerobic microbes, microorganisms with some, for example, a fermentative Clostridium, and they were all related with environmental sequences from lake sediments, permafrost, periglacial environments, and northern peatlands. When we do the same analysis only with the methanogenic community structure, what we found is that the most important was the methanogenic precursor that we added. So it was expected, actually, because we were expected an adaptation of the community, and that's the case. So here, as we saw before, the methanobacterium became dominant in the hydrogenotrophic experiments, and what was also interesting is that some acetate has been produced during these experiments by this specific acetobacterium. So what we observed in these experiments that the first substrate that we added was first consumed by acetobacterium producing acetate, and then it seems that acetate was then transformed again into hydrogen to produce then methane and to give this substrate to the methanogens. So it was really dynamic in this microcosm there. In the acetate-ammontid microcosms, we observed a dominance or a selection of methanothercina. Here, for example, this O2 was more affiliated with methanogen that can use both acetate and methanol, and found in permafrost and lake sediments. Actually, with no substrate addition, we found important this metanobacterial O2, which is a methanol linear. It's an hydrogenotrophic methanogen, and what is interesting here in this experiment from without any addition is that we found non-methanogenic archaea from the group of marine benthic group D. And what was interesting is that these archaea can produce acetate and ethanol through fermentation, so they can give or they can provide a substrate for the methanogens then. So actually we, in that microcosm experiment, in that enrichment, we found that increasing temperature can increase the microbial activity and the methane production in that case. What we found is that acetates seem to be more efficient in stimulating methane production in that kind of ecosystems. And interestingly, the hydrogenotrophic based methane production rates were more affected by temperature increase. Even at this low temperature, I mean that methane or methanogenesis has an optimum at 37 degrees. So here for methanogenesis, we were working at low temperature between 5 and 20. And what we found also is that biogeography of microbial diversity is needed and really important, since even after incubation, the ecosystem of the origin of each microbial community was still important and still relevant. Okay, so then what we realized is that actually the methane cycle can also be linked to other cycles, to other biogeochemical cycles. And for example, Mercury cycle can be an associated cycle with the methane one because of mainly of methanogenic archaea actually. And we were highly interested in studying the implication of these microorganisms in aquatic ecosystems, and particularly focusing on Mercury cycle. So let's go to Antarctica now. And why we were studying Mercury? Because South America is responsible of 18% of the anthropogenic mercury emissions. And that's one of the reasons why Chile ratified the Minamata Convention more than 2 years ago now. And also because the problem in Chile here is the mercury emission and mercury release from coal mining industry. And most of that, the problem of mercury is that mercury can be methylate into methyl mercury, which is an important neurotoxin that could first bioconcentrate from the water to the organisms, then bioamplified all along the life of the organisms, and then biomagnified along the trophic network. So it's an important issue here in Chile, but all around the world. And if methylation can occur through abiotic processes, microorganisms are also implicated in that process, and we still don't really know and don't really understand at which proportion microbial processes can be important here, and we don't really know in different kind of ecosystem what is the part of the methyl mercury produced by microorganisms. So we have experimental proof of many anaerobic organisms for methylation, but we also suppose, thanks to molecular data, that other organisms, including aerobic organisms, can also have the possibility to methylate the mercury. And what was interesting, what is interesting for us also is the demethylation of methyl mercury. Well, actually, this process is interesting, and we wanted to understand why the link with the methane cycle. We are still working on that. So we are first trying to understand the basis, because we don't have a lot of data in these known ecosystems. So we are actually working on that, and what I am presenting now is like preliminary work that we are doing, and we hope to be able to give more results as soon as possible. So let's travel to the south and to the King George Island at the front of the Antarctic Peninsula. Here you have a picture in summer, and it was in February, I guess. And why are we studying these aquatic ecosystems? Because mercury can be naturally present there because of the volcanism origin of the substrate. But mercury is also transported by long-range atmospheric transport and can be stored in different freshwater ecosystems, including glacier, and can be also released during the melting season. So it's important for us to understand what's happening during the melting season with the mercury cycle, and what's a proportion or what is the importance of the microorganism there. So here I presented the result from 2 different ecosystems. The first is the Kittish Lake, that is a lake on the King George Island with a maximum depth of 10 meters, and a little glacial pond here in a little island at the front of the King George Island. It's a very shallow pond there. So we studied both in sediment and in water the total mercury concentration and the methyl mercury concentration. We also associated some 16S metabar coding analysis, both targeting both at the DNA level and the RNA level, and we make some methanogenic analysis, and we couple that with isotopic enrichment in order to measure the mercury methylation in these ecosystems. What we found is that actually we found mercury, and we found methyl mercury in water also. We still don't have the results of the methyl mercury in sediments, but it's coming soon. What we found is that until 30% of the mercury that we found in water was actually methyl mercury. And interestingly, in the Kittish Lake sediments, we found total mercury higher than the value of the baseline established before in that island. Actually, the mercury concentration are pretty low, but higher than the baseline. We also measure the methylation rate. So here we can find that after 24 hours, the methylation rates were pretty similar between the two ecosystems. And if we look at the putative methylating microorganisms there, what we found is that they were more abundant in sediments, and that was expected, and more diverse in sediments also. We found an importance in sediments of the Chloroflexi for example, and in water we found nitrospira, which was recently found to exhibit these marker gene of the methylation, which is the HGCA. Looking at the metagenomics data, actually we are still analyzing this data. We were able to collect more than 220 marks, and we found both MERB and HGCA, so both marker of demethylation and methylation of mercury in the metagenomes. And actually, if I use a similar figure that Poda and collaborators have done in their very interesting study, we found that here in the current study, the abundance here in color, the abundance of HGCA gene normalized by the metagenome size was really high compared to other ecosystems, for example, in Wisconsin or near Seattle. So in these Antarctic ecosystems, we finally found that until 30% of the mercury in water was methyl mercury. We found the presence and putative activity of methylated bacteria using molecular data. And we obtained a lot of metagenomes that we will, a lot of mags from the different metagenomes we studied, including exhibiting these marker genes. And we also gave the proof of the activity of these organisms with isotope techniques. Actually, these isotope techniques are really sensitive, and we are still performing some analysis, or more analysis actually, because comparing the value of mercury methylation is pretty hard because of the protocol. So with French colleagues, they are trying to develop a new protocol, or a uniform protocol, in order to be able to compare all the data according to the different ecosystems. So this is still a work in progress. And we define that there is a necessity to improve our knowledge on the microbial mercury dynamic there in the south, and in these kinds of ecosystems. Here, picture before to conclude from that kind of study. Well, actually, if we try to conclude with a global point of view, what I consider is that in a context of climate, of global warming, of melting, higher melting or thawing of glacier and permafrost, it is supposed to be to have an impact on greenhouse gas emission and on contaminant releases, and microorganisms are a key compartment to consider in that kind of processes at the Earth scale, because they can have an importance at that scale. And actually, I consider that studying this unique ecosystem from the south gives the opportunity to understand better global warming. We know that the temperature are increasing really fast in the north, and it seems to be slower in the south still at that moment. And so we still have the opportunity to understand better what can happen in increasing temperature scenario, and what's happening in melting season, for example, when the contaminants are released from the glacier or from the cryosphere. So studying the south can allow to anticipate and understand the effect of these anthropic activities. Actually, I would like to say that if you are interested in that kind of studies, we are offering a PhD now in the frame of a new project that we are beginning. We are interested in understand the role of bacteria and archaea in mercury transformation in aquatic systems, but in the Andes Cordillera, so in the glacier. And we want to study that from the glacier to the sea during the melting season. It's supposed to be a start in August of this year and take place in Chile. So if you are interested in doing a PhD here in Chile, just feel free to write me an email. It will be a pleasure to discuss with that. And before finished, I wanted to tell you about a new initiative that we've built with 16 other women researchers. We created last year the first Chilean network of microbial ecology. It's called RECHEM. We have a Twitter, we will have a website also. And actually we are 17 researchers, but we join or we put together more than 500 people interested in microbial ecology in Chile and internationally also. And just to give an idea of what we have done, actually we have an initiative here we built in collaboration with the designer and microbiologist Alexa Garin Fernandez from the Max Planck Institute. We built the first microbial map of Chile. So what the idea was to report here the microorganisms that were first cultivated here in Chile, and to put it in the map. Here it's in Spanish, but we also, in the website, we also have the English version if you are interested in. So, well, actually, I wanted to thank all the people working with me, and I think I'm sorry if I forget someone. I also want to thank all the fundings, because, for example, the Chilean Antarctic Institute is doing an amazing work to let us study the Antarctic ecosystems. I went to Antarctica 2 times for this kind of studies, and it's an amazing place to do research. The education, or now the science Ministry of Chile also is providing different fellowship or fundings. So that's a very, very nice opportunities, and thank you to all the people that are working with that. And well, actually, thank you for your attention. Sorry for my French, Spanish, English, but anyway, I will be pleased to answer some question or begin some discussion. Thank you.
Thank you very much. That was great. Okay. Well, we have a few questions in the chat already, so I'll pass those on from Kokle Clement, I hope I'm pronouncing that correctly, as well as Roland Hotzenpickler. Basically a similar question. How do you identify the methanogenic taxa? And more specifically from Roland, are you using MCRA or 16S alone?
Yes, I did not say that. Actually, in that data, I used the 16S. So I used the known taxa and I searched it in the database, in the 16S database. I also quantified the MCRA, but I did not sequence the MCRA in these samples.
Okay, and another question from Roland. He also said, thanks for the talk. I was wondering whether you looked at use of methylated compounds in your methane incubations. Would these compounds, methylamines, methyl sulfide, etc., be present in situ?
Yeah, so actually we were focusing on these two other methanogenic precursor. I get that this is the question, and why I don't add methylated compounds. Actually, I did not measure the methylated compounds in these samples. So it's an important point that we hope to be able to improve for the next experiment. But actually, we did not. What we did is that in some samples we have isotopic data of methane, and thanks to this data, we established that the most putative pathway was the acetoclastic pathway, looking at the isotopic data.
Interesting. Let's see, follow-up question from Roland. He says, thanks. Actually, this is a comment. Thanks, just be careful with using only 16S. 16S doesn't assemble well, and not all genera that contain methanogens do only methanogenesis. See, for example, a recent paper by Laura Zinke, EM 2021, and also true for Moth Bathy and Verstrate Archaeota. So I guess that's just a warning.
Yeah, but that's okay, because yes, the 16S is not enough, particularly for function. It was the one available actually in the data set, but I understand the comment.
Yeah, we're on a little bit of a lag, so I'll ask a question and see if he wants to follow up. I'm curious, because I know you were very interested in understanding, not a lot of work has been done on like the methanogens down in southern hemisphere compared to like northern hemisphere, and then of course you got all these mercury mercury active or mercury metabolizing organisms that you've got mags for. How similar, or do you know yet how similar those taxa are to existing known taxa? Like you found these Nitrospira down in Antarctica. Are those similar to the Nitrospira that have been previously characterized, or are we talking like, I guess what I'm getting at here is, can you tell whether we're looking at basically the same types of organisms that are spread throughout the globe, or if these look like they may be slightly different versions, slightly evolved, maybe different species, different genera, something like that?
Well, actually, that's a question that we have, because actually the putative methylator Nitrospira was found mainly in marine ecosystem and in the water column of the oceans. And we actually don't have the answer yet, because we are still analyzing the mags. So I hope to find a Nitrospira mag and be able to answer to that question.
Cool. Okay, Great. We have another question. Roland also said to say thank you for the answer from username Paul ACJR. Do you think methanotrophy in these southern aquatic ecosystems has the potential to offset the increase in methane production caused by increasing temperatures? And he also wants clarification, are these lakes stratified or mixed?
Well, actually, these lakes are quite shallow and they are mixed. Depending on, depending on the season, but in that case, we only work with one lake, with one stratified lake. The glacial lake actually near Punta Arenas was stratified, and the beginning of the question was if I did not get it.
Sorry, I'll repeat. Yeah. Do you think that methanotrophy in these southern aquatic ecosystems has the potential to offset the increase in methane production caused by increasing temperatures?
Well, actually, I did not present that, but we have emission data because in the frame of the project, we also work with amazing colleagues working in biogeochemistry. So we know that in these ecosystems, the total emission was positive. So these lakes were emitting positive quantity of methane. And so we support that. We didn't still calculate the balance, but it seemed that the methanotrophy within the water column was not sufficient to fully oxidize all the produced methane by produced in the sediments.
Interesting. Great. Okay, well, this was a wonderful contribution. There's no more questions at this time, but keep checking back. And those of you who are listening, if you come up with questions now, or if you're watching this recorded sometime in the future, please get in touch with Dr. Lavergne. And just to remind everyone, there's a wonderful PhD opportunity in her lab. So if you're looking for PhDs
please.
right now and aren't shy about going to Antarctica, which sounds incredible, get in touch.
Thank you. Actually, if I can just add something, is that
of course.
I hope that Chile's government will still propose a postdoc fellowship. So you just have to apply, propose the subject, and get it. This year it was not possible because of the pandemic, but we still have a lot of metagenomes to analyze. So if someone is interested in help with that during a postdoc with us, please, you're welcome also.
Wonderful. Okay, some great opportunities there. Well, thank you very much, Dr. Lavergne. And for everyone who's listening at home, have a good rest of your week. Bye-bye, everybody.
Bye, thank you.